Drug effects on postsynaptic mechanisms. Effects of neurotransmitter receptors. One is the blockade of receptors. An example are antipsychotic drugs block dopamine D2 receptors, and Curare blocks nicotine acetylcholine receptors. Activation of receptors. Example, nicotine activates acetylcholine receptors. LSD is an agonist at some types of serotonin receptors. B. The effects on cellular process. First is the regulation of the number of postsynaptic receptors. An example is alcohol regulates or increases the numbers of receptors for GABA. Another example is modulation of intercellular signals. An example of that is mood stabilizer lithium has many effects, including changes in second messengers, probably leading to changes in gene expression and receptor density. Some neuroactive drugs ease the symptoms of injury or psychiatric illness. Antipsychotics relieve schizophrenia. A majority of institutionalized psychiatric patients suffered from schizophrenia pre-1950s. Antipsychotics, or neuroleptics, is a family of drugs that suddenly and dramatically improved the schizophrenia situation. Chlorpromazine, haloperidol, and loxapine are all examples of antipsychotics. All of these drugs act as selective antagonists of dopamine D2 receptors in the brain, and all were typical antipsychotics at first. They were very good at relieving the positive symptoms of schizophrenia, like hallucinations and delusions. And a dopaminergic model of this disease became dominant because these drugs were so successful. The 1990s was the advent of the second generation schizophrenia medications. These are called atypical antipsychotics. They have both dopaminergic activity and additional non-dopaminergic actions. This includes a blockade of certain serotonin receptors. Clozapine is an example of this drug, and it reduced the negative symptoms of schizophrenia that were resistant to typical antipsychotics. This includes impairment or loss of behavior, social withdrawal, and blended emotional responses. Evidence that schizophrenia involves additional non-monoaminergic mechanisms has prompted an intense research effort to help develop third-generation antipsychotics with novel targets like glutamatergic synapses. Effective treatments remain elusive. Schizophrenia remains a difficult, multifaceted disease and a major health problem. Antidepressants reduce chronic mood problems. Affective disorders, disturbances in mood, most common of all psychiatric complaints. Antidepressants occurred at first in the 1950s, and these were monoamine oxidase inhibitors, or MAO inhibitors. An example of it is tranylcypromine and isocarboxazid. MAOs normally break down monoamine neurotransmitters at axon terminals, reducing transmitter activity. MAO inhibitors block this process and allow monoamine neurotransmitters to accumulate at the synapses. The result of this drug is an improved mood. Increasing synaptic monoamine availability is the key activity in all antidepressants. The second generation of antidepressants are tricyclics, and they combat depression by increasing the synaptic content of the monoamines, norepinephrine, and serotonin. They are three-ring molecular structure, and they block the reuptake of neurotransmitters into the presynaptic axon terminals, allowing the transmitters to accumulate in the synapse. An example of it is imipramine. Recently developed are SSRIs and SNRIs, and they alleviate depression by selectively allowing serotonin and norepinephrine with SNRIs to accumulate in synapses. They lack undesirable side effects of older drugs, but they still have new side effects. This includes disturbances of sexual function, and they take 6 to 8 weeks to have full effect. Anxiolytics combat anxiety. Clinical states of anxiety include stricken by disabling emotional distress that resembles abject fear and terror. This includes panic attacks, phobias, generalized anxiety. Anxiolytics are used to treat these clinical states of anxiety. They're also called tranquilizers, and they're in the general category of depressants, or drugs that depress or reduce nervous system activity. Alcohol and opiates are probably the original anxiolytics. Anxiety fighting comes at a cost of intoxication, addiction potential, and neuropsychological impairment with long-term abuse, so they are not suitable for therapeutic use. But barbiturates, or downers, originally developed to reduce anxiety, promote sleep, and prevent epileptic seizures. And they're still used for these purposes, but they're very addictive and easily overdosed. Benzodiazepine agonists are most widely prescribed anxiolytics since the 70s, and they're more specific and safer than barbiturates. Example is diazepam, Valium, or lorazepam, Ativan. They bind to specific sites on GABA A receptors and enhance the activity of GABA. GABA A receptors are inhibitory, so benzodiazepine helps GABA produce larger inhibitory postsynaptic potentials caused by GABA alone. And it has an end result of reducing excitability of neurons. GABA A receptors have several binding sites. Some facilitate and some inhibit the effects of GABA. Many different drugs can interact with this receptor complex. Benzodiazepines bind to a unique modulatory site on the receptor complex that is distant from where GABA itself binds. And the benzo binding site is an orphan receptor, which means it is a receptor for which an endogenous ligand has not been conclusively identified. Allopregnanolone is a steroid developed from hormone progesterone, and it acts on another site on the GABA A receptor. It's elevated during stress and has a calming effect. And alcohol ingestion increases brain concentrations of allopregnanolone, so it might mediate some of the calming influence of alcohol. Progesterone, like steroids produced in the brain, may act on GABA A receptors to produce anxiolytic, analgesic, and anticonvulsant effects. The GABA receptor has 5 protein subunits that penetrate the cell membrane and surround a chloride ion channel at the core. Many depressant drugs work by increasing the responsiveness of GABA A receptors to GABA. Opiates potently relieve pain. Opium is extracted from poppy flower seed pods, and it's used by humans since the Stone Age. Morphine is a major active substance in opium, and it's very effective analgesic or painkiller, and has brought relief from severe pain to many millions, but it has a very strong potential for addiction. Heroin, or diacetylmorphine, is a relative of morphine and is also very addictive. Opiate painkillers include oxycodone and fentanyl, and there have been accidental overdoses of fentanyl commonly throughout the world. Opiates like morphine, heroin, and codeine bind to specific receptors, opioid receptors, concentrated in certain regions of the brain. They're found in the limbic and hypothalamic areas of the brain. They're particularly rich in the locus coeruleus and the periaqueductal gray matter that surrounds the cerebral aqueduct in the brainstem. Injection of morphine into the periaqueductal gray produces strong analgesia, indicating that this is a region where morphine acts to reduce pain perception. There are 3 major families of endogenous opioids, which include enkephalins, endorphins, and dynorphins. And there are 3 main kinds of opioid receptors, and they're all metabotropic receptors. The first is delta, second is kappa, and the third is mu. Powerful drugs that block opiate receptors can rapidly reverse the effects of opiates and rescue people with overdoses. Some neuroactive drugs are used to alter conscious experiences. Cannabinoids have a wide array of effects. Marijuana is derived from cannabis sativa plant and is used in various cultures. It contains dozens of active ingredients like THC. The use of marijuana usually produces pleasant relaxation and mood alteration. Heavy use can be harmful. It's mostly associated with respiratory problems, addiction, psychiatric disorders, and cognitive decline. Adolescents that use marijuana appear to be more likely to develop psychosis in adults, but it's still unclear if the drug causes it or if the pre-psychotics are more likely to turn to marijuana. The brain contains cannabinoid receptors that mediate effects of compounds like THC, and they are concentrated in the substantia nigra, hippocampus, cerebellar complex, and the cerebral cortex. There are at least two subtypes of cannabinoid receptors, CB1 and CB2, and they're both G protein coupled metabotropic receptors. CB1 is found mostly in the nervous system, and CB2 is found in the immune system. Endocannabinoids are endogenous ligands for the cannabinoid receptors, and they function as retrograde messengers. They convey messages from the postsynaptic cell to the presynaptic cell, and it's a retrograde signal is thought to modulate the release of neurotransmitters by the presynaptic nerve terminal. Anandamide is the most studied endocannabinoid, and it has diverse functional effects like alterations of memory formation, appetite stimulation, reduced sensitivity to pain, and protects from excitotoxic brain damage. Most CB1 receptors are found on the GABAergic axon terminals. So yeah, that's segment 3 of chapter 4.